Good morning, students. Today we'll discuss about the immunoglobulin G. Immunoglobulin G is a large monomeric molecule of about 150 kilodalton. It has two chains, heavy chain and light chain. Heavy chain is also known as the gamma heavy chain. These two heavy chains, they are having a hinge region in their constant region. Each heavy chain is having two types of region. Variable region known as VH, constant region, they are known as CH1, CH2 and CH3. Whereas the light chain is having only two regions, that is the VL and CL. CL is the constant region, VL is the variable region. The fork-like end, which is formed by the immunoglobulin G, is known as Fab region, whereas the end with which it is attached with the other cells is known as FC region. The light chain may be consisting of kappa chain or lambda chain. IgG is the most abundant type of antibody found in all body fluids and protein against bacterial and viral infections. It represents approximately 75% of the serum antibodies in the human. It has the serum concentration of 10 to 16 mg per ml and is also considered as major immunoglobulin in extravascular spaces. The IgG antibody elicit response such as antibody-dependent cellular cytotoxicity, antibody-dependent cellular phagocytosis, as well as complement-mediated cytotoxicity. IgG is a main type of antibody found in the blood and extracellular fluid, allowing it to control infection in the body tissues. The binding of Fc portion of IgG to the receptor present on the phagocyte is a critical step in opsonization. Phagocytosis of particle coated with IgG antibody is a vital mechanism that cell uses to cope with microorganisms. So in this animation you can see that the macrophages, they are consuming the bacteria with the help of the antibody bound or antibody coated. Secondly, in the case of the second diagram, you can see the receptor CRI is present with which the antibodies, they are bind and ultimately the bacteria, it is consumed. IgG is the main type of antibody found in the blood and extracellular fluid, allowing it to control infection of body tissues. IgG is the only class of immunoglobulin that can cross the placenta in humans and is largely responsible for protection of newborn during first month of life. IgG is a major immunoglobulin in blood, lymph fluid, cerebrospinal fluid, and peritoneal fluid and is a key player in the humoral immune response. So in this way, IgG can cross the placenta. IgG is produced in a delayed response to the infection and can be retained in the body for a long time. The longevity in serum makes IgG most useful for passive immunization by transfer of its antibody. Detection of IgG usually indicates prior infection or vaccination. Because of its relative abundance and excellent specificity towards antigen, it is a principal antibody used in immunological research and clinical diagnosis. There are four types of IgG subclasses: IgG1, IgG2, IgG3 and IgG4 in humans, mainly based on the order of their abundance in the serum. That is, IgG1 is most abundant. So these subclasses, they are differentiated on the basis of size of the hinge region. IgG3 has longest hinge region. Position of interchain disulfide bonds and molecular weight. The subclasses also differ in their ability to activate the complement system. So IgG2 cannot cross placenta and has low affinity to bind to Fc receptor of phagocyte, whereas IgG4 does not activate complement system. Others they can. So in this animation you can see how the IgG is responsible for causing the phagocyte to consume the cell, for causing the NK cells to cause the cell lysis, as well as bringing the antigen towards the phagocyte, and ultimately leads to the digestion of this phagocyte with the help of the phagosome and activation of the complement system with the help of IgG, leading to the formation of MAC, that is membrane attacking complex, and thereby causing the cell lysis. So to sum up, we can say IgG is responsible for neutralization of microbes and toxins. IgG is responsible for opsonization and phagocytosis of microbes. IgG dependent cellular cytotoxicity can be there with the help of NK cells. Phagocytosis of microbe opsonized with complement fragments, that is C3b, is also there. Then IgG, when it activates the complement system, it can lead to inflammation and lysis of the microbes. Clinical implication of IgG. The increase in IgG can be indicated of the infection, liver-related diseases, severe malnutrition, hyperimmunization, rheumatoid arthritis, and dermatological disorders. I hope the concept is clear to you regarding IgG. Thank you.